Welcome to The Mind, the Cure Protocol for July 27, 2026. No newly verified patient reaching milestone in this pass has reached care, so we will not pretend otherwise. We are following what changed in the evidence, what new research it opens, and what still needs proof before it can help anyone. This protocol is a living loop. People bring the questions that matter. Fresh evidence changes the map. The system remembers what we have already seen, so an old announcement does not keep pretending to be new. Every signal ends with one practical next action, the experiment, validation, collaboration, or care decision that could move it closer to real benefit. Researchers in this study used AI-driven multi-omics integration and virtual screening to identify UFC1 as a druggable effector in chemotherapy-stressed colorectal cancer, linking copy number amplification and elevated expression to chemotherapy response. By turning heterogeneous tumor data into a mechanistic target, the work illustrates the broader AI-for disease shift, intelligence inversion from trial and error biology to computational prioritization, and a phase transition in which cancer resistance becomes an engineering problem of finding the control nodes. If confirmed, this could help clinicians see disease signals more clearly and focus the next decision. The useful part is precision, not magic. A clever sensor still has to prove it helps a real clinic make a better decision. Next, show in independent studies that the measurement improves a real clinical decision. Locus Biosciences announced a $3.3 million federally backed clinical trial of an AI-designed bacteriophage therapy for hospital-acquired and ventilator-associated pneumonia caused by antibiotic-resistant Pseudomonas aeruginosa. If confirmed, this could give antibiotic teams a better starting point against resistant infections. The useful part is a more practical route to treatment. The chemistry still has to survive scale, safety, and real bacteria. Next, reproduce and scale the chemistry, then test the antibiotic against resistant bacteria. Pfizer and its partners reported that Sonesitatug vedotin produced a statistically significant, clinically meaningful overall survival benefit in CLDN 18.2 positive advanced gastric-cancer after second-line therapy, with the Clarity's gastric-01 data also supporting expansion of the CLDN 18.2 positive definition to greater than or equal to 25% expression. If confirmed, this could help clinicians see disease signals more clearly and focus the next decision. The useful part is precision, not magic. A clever sensor still has to prove it helps a real clinic make a better decision. Next, show in independent studies that the measurement improves a real clinical decision. Researchers behind scGPT and Geneformer have shown that transformer-based foundation models can learn a cross-cell language from single-cell RNA sequencing data, enabling prediction of cell state, perturbation response, and biological annotation at scale. If this holds up, it could give researchers a clearer target for the next experiment, validation step, or collaboration. The useful part is a clearer target, not a finished solution. Independent evidence still has to do the hard work. The next question is whether independent evidence confirms the finding and shows a responsible path toward real-world use. The practical read is simple. These are signals, not promises. But each one is a concrete target that researchers, clinicians, engineers, funders, and the people most affected can connect to a next step. Keeping the evidence visible is how the Cure protocol stays current without pretending the work is finished.